Oh hey, didn't see you there. This might seem like a pretty unassuming hallway. There's labs, offices, even a classroom down the hall. But in this room right here, there lies the most concentrated fungal diversity in the world. The Amazon rainforest, the Pacific Northwest, even your bath mat. Take any room-sized area they're in, and they'll all pale in comparison to what's in here. So let's go in and take a look. This is a fungal herbarium. Herbaria are collections of natural specimens, sort of like libraries for the natural world. Inside of these cabinets lies boxes upon boxes of specimens, some of which date hundreds of years back. In this little area, nearly 10,000 fungal specimens are stored and maintained. There's less than 200 places like this in the world. And places that have one of every mushroom? It's every forager's dream. But one question may come to mind when seeing all of this. Why? What's the actual value of these facilities, and what do they actually do? I'll be touring the Larry F. Grand Mycological Herbarium to learn about Herbaria and answer all of your questions and more. I hope you enjoy. Today I'll be chatting with the herbarium's current curator, Andrew Paul. I'll be interjecting with some commentary intermittently. If you like these sort of interview style videos, let me know down in the comments. Hello, Andrew. Thanks for sitting down with me today. I guess first up, could you describe what's the purpose of an herbarium?
Sure. An herbarium is a collection of dried plant or fungal specimens that is preserved for future research. So we preserve our collections. We don't know, maybe don't know what to do with them right now, but in the future they might be useful to someone else. So we keep them around in good condition so that people can look at them.
Some great insight into what an herbarium actually does. Historically, herbaria have served as reference material. When you find a new species, you have to document it. I got Andrew to show me my favorite group of fungi, Craterellus. Get the name now? If you look online for the specific epithet of one of these species, let's do this one, Craterellus cornucopioides, you'll see next to it or below it, L. pers. Back in 1753, that L guy, Carl Linnaeus, found something much like this, documented all the features he could, and named it Pisiza cornucopioides. He wrote a paper talking about this new species, the purpose being, this is mine, I discovered it first. Because he discovered it, his name stays there forever. The original fungus was dried and labeled as the holotype, or simply type. This type is kept in herbaria and is used as the standard for comparison for all other specimens of the species. If all goes well, it stays there forever. But this guy Christian Persoon came along in 1825 and was like, for these scientific reasons, this species should actually go in a different genus. And since nobody else has been successful in dethroning Persoon, trust me, many have tried, his name stays right there. But not every single fungus in an herbarium is a type. In fact, the vast majority aren't. These others are records, proof that the fungus existed at a certain place and time. These are collected in ecological studies and surveys. They have uses beyond comparison that Andrew will detail soon. So that's great. Now we have a reference for fungal species in these type specimens, as well as a physical record for historical specimens. What do you do as the curator of the herbarium? What's your day-to-day?
As the curator, I'm in charge of adding specimens to the collection. So when I receive something that someone has collected, I first freeze it so that there's no insects that'll destroy it, and we don't want to introduce insects into our collection. And then I dry it so that it'll last a lot longer. So my day-to-day looks like evaluating these specimens and adding them to our database with all the good information, so who collected it, where was it collected, and then adding it to our collection in order so that it can be found later.
And I guess that's a great segue. What does your research entail?
So our research looks like describing new species or going into the collections to see if the old species had the correct name. So for someone else says it is a new name, then I go and check that it matches what they said it is. So I'll take a specimen and look at it under the microscope and take photos of that, and also research the range where it is found in the world and previous collections in other herbaria around the world.
Dr. Michael Bradshaw is the herbarium's current director, but he has to split his time between the herbarium with his lab, the Bradshaw Lab. There he studies a group of plant pathogens known as powdery mildew, which, I'm not gonna lie, they're kind of boring. At least that's what I first thought. I was cynical. These little white spots? That's it? But Dr. Bradshaw's enthusiasm, much like the fungi he studies, is contagious. Obviously, Dr. Bradshaw has his lab. Is herbarium material included in his research?
Yeah, absolutely. So Dr. Bradshaw's lab studies powdery mildew, which is a important plant pathogen on lots of economically important crops and just ecologically significant species around the world. So part of our work is requesting other herbaria to send their powdery mildew collections to us. We're trying to build up our collection because that's the focus of the lab, but it also looks like sequencing the DNA that we can extract from the specimens we have here and from historical specimens, and we'll be able to get DNA with a positive identification off of species that are over 100 years old.
Dr. Bradshaw is currently running a project to document the diversity of North American Irisaceae species, powdery mildew. This research will help improve identification and treatment of the disease, and I can say I've come around to the modest mildew. But obviously, Dr. Bradshaw's group isn't the only group doing great research with herbaria. What are some interesting ways researchers like yourself utilize herbarium material?
So what's really interesting about herbaria is they kind of add this element of time to your research. So you're looking at specimens collected in the modern day and also in the past. So you can track how species have changed, where they're located, and especially with like invasive species. If you've never seen a specimen somewhere, then all of a sudden you're collecting it a whole bunch. You can kind of track that, decades after it happened. You can find that evidence of invasion. Also with climate change, as certain areas get warmer, you can see that species are found in new locations where they weren't found previously. But it's especially important with diseases. We're tracking where they might be found and where they might be found in the future to help predict who might be impacted.
I'm hoping to go into research one day, and this is all super cool. Almost as cool as you hitting the like and subscribe button. Ahaha. No, seriously, thanks. It really helps. Despite my ramblings about research and the like, there's a question I had to ask to itch that forager side of me. What's the coolest fungal specimen you have?
What I think is the most interesting is we have collections going back like decades or even longer. So I think probably one of the oldest specimens we have that we can still get important research done on it. It's probably one of my favorites.
Can we see it?
Yeah, for sure.
Right,
let's take a look.
Let's take a look. All right. Dried mushrooms are stored in boxes like this one, but that's not all. Plant pathogens can also be stored in envelopes. Packets of material with leaves, branches, and more can all be found in the herbarium. Given Dr. Bradshaw's interest in powdery mildews, it's no surprise that he's amassed quite the collection. Each of those red lines is actually a type specimen. Drying is a crucial step to ensure unwanted fungi doesn't grow, as well as to prevent insects from enjoying the fungal feast. Before today's systems of imaging and easy worldwide shipping, there existed these sort of scrapbooks of material known as hexacadi. Shared amongst herbaria, they contain some of these type specimens and made it easy to compare common species across the world. Most pages had multiple species, and when hexacadi became outdated, they weren't thrown out, they were archived. At herbaria, hexacadi were dissected into individual species and remounted onto new paper. A single book became an entire cabinet's worth of specimens here at Larry F. Grand. Now on to what I believe is the most controversial question when it comes to fungal herbaria. There's a good chance the interview ends here right now. Is it fungal herbarium or fungarium?
I personally prefer mycological herbarium because I think the term herbarium was around first, and then they started collecting fungus, and I think fungarium is a little bit, it's not as good of a word. People don't know what you're talking about if you just say fungarium.
Okay, I lied a little bit. Historically, fungi were considered plants, so they were put in plant herbaria, but since we found out they're not plants, mycological herbarium, fungal herbarium, or fungarium will all generally work. Now on to a bit of information about me. Why am I even talking about this? I've worked at a plant herbarium for a couple of years now, and working there has really shaped my views on conservation and the roles these records can play in research. It's really a marvel that thousands, if not tens of thousands of people have played roles in this whole crazy system we have. From the original collectors to people who process and identify samples, people like me who mount and sort them, people like Andrew who are researchers who work with these samples and upkeep them. That is to say, there is a lot that goes into these collections, which makes what I'm about to say all the sadder. In February of 2024, the decision was made by Duke administration to shut down the Duke Herbarium, plant and fungal. Duke has an immense collection of plants and fungi, with over 825,000 plant specimens and over 14,500 fungal ones. It's the second largest private herbarium in the country, just after Harvard, focusing on the fungi, which are mainly basidiomycetes, i.e. mushroom-forming fungi. The NC State Herbarium is able to hold 8,500 specimens because many of them are plant pathogens. Again, they fit in these envelopes, which takes up a lot less space than these relatively larger boxes which the mushrooms are held in. There are 20,000 signatures on change.org, appeals from former herbarium staff, the Mycological Society of America, the Society of Herbarium Curators, Science Magazine, and a 7-page long list of others who oppose the decision, and for good reason. They plan on relocating much of the herbarium, but with a collection so big, there are challenges. What are your thoughts on some of the recent herbarium closures?
I think it's really detrimental to the progress of science looking into the future, where recently the Duke Herbarium, which was a close collaborator with us, it recently closed, and there's the important part of Herbaria is the physical collections, where you might be able to digitize them in some way, but it won't be the same as having that actual specimen there where you can manipulate it and get DNA off of it. And the whole point is that we're preserving them into the future so that even if we don't know what to do with them now, someone else in the future will have an idea and they'll be around for them to use. And now it's kind of up in the air where those specimens will be once the herbarium closes.
Closures like these have a chilling effect. If the second largest private herbarium in the country can have its funding cut, who might be next? In an academic environment which doesn't favor such work, it can seem pretty depressing, but there are people who continue to work to save these specimens, and fortunately, there's some hope. Other herbaria have received massive support in recent years. The Missouri Botanical Gardens received an anonymous multi-million-dollar donation to help support their herbarium. Universities like Iowa State and those close to Duke, UNC Chapel Hill and NC State, are stepping up to face the challenge, and luckily, you can too. How can citizen scientists or volunteers help with fungal herbaria such as this one?
Anyone that's out in the woods or out anywhere even can find a specimen, and if they think it's something interesting and they want to find out more about it, they can take it to us. Recently, a local teenager gave us a sample of Ophiocordyceps, which is the fungus that grows on ants and takes them over. They just found an ant that looked like it was infected in the woods, and now I accessioned it and put it in our collection under his name, so he can always come back and be immortalized in the herbarium. But anyone that wants to collect and wants to learn more about fungus can come to your herbarium and either add to the collection or help you identify that and just improve what we know about these specimens.
With already limited funding potentially being more limited by funding cuts, there's ample opportunity for citizen scientists and volunteers to help preserve and support these collections. Alongside collecting, quite a bit of volunteer work is sometimes needed for some of these collections. In fact, there are outlets to do so. MycoPortal is an online database that contains all of the digitized specimens from fungal herbaria. Many of these specimens have physical data on the sheet, but that hasn't yet been translated to digital. You can help reduce the workload on scientists simply by going online and transcribing. The MycoPortal website has a guide on how to do it and a leaderboard, and if you try hard enough, you might be able to beat the number one, Kevin Healy. Find a local mycology club and ask about connected researchers. If you're in high school or college, see if you have a local herbarium and contact them. Herbarium curators and directors are busy people, but they're often more than happy to receive some extra help. I myself have been greatly impacted by herbaria, and I highly recommend that you volunteer at one, plant, fungal, or otherwise. If you're passionate enough, you too can become a protector of the fungal past and a steward for the fungal future. Huge thanks to Andrew and Dr. Bradshaw, who are truly amazing people and answered all my questions like pros. And of course, thanks to you for watching my video and supporting my content. I'm hoping to make more interview-style content, so if that floats your boat, let me know by liking, subscribing, and leave a comment while you're at it. Thanks, I truly appreciate it, and as always, take care.